Hey friends, welcome back to Crunch Academy. In the previous sessions, we learned about the generation of action potentials, how are they being transmitted across a neuron, and in the last video, I did promise you that we'll be learning about how this information from the neurons reach the muscles, or how are neurons able to transmit their signals to the muscles. Okay, so consider this to be my spinal cord. Okay, I'm taking a cross section of this. Okay, and I'm viewing it from the above, and it looks like this. Okay, with the central canal. Now this looks like horns, right? So, and since when looking from the above, these are the posterior part and these are the anterior part, we refer these two horns as anterior horns, and we refer these two as the posterior horns. Okay, now what is specialty about anterior horns? We learned that in the central nervous system, the sensory system is responsible for carrying information from the body and around the environment to the brain, and the brain processes them, and signals from the brain are taken to the muscles or the target organs or the effectors by motor neurons. So consider this to be our cross section of our spinal cord, with this being the anterior horns. Okay, or they can also be called the anterior horn cells. Okay, and within this anterior horn cells, who lies? Our cell bodies of motor neurons. Okay, now consider this to be our neuron. We have our cell body part with the nucleus, and we have the axon, and we have the synaptic end. Okay, this cell part of the neuron, which is a cell body, is embedded within the anterior horn cells, and the rest of the part is what I have zoomed here. Okay, that is from the axon. This is the axon, this is the axon, and this is the nerve ending. Okay, now what are these? This is, consider this to be a muscle. Okay, and the part of the muscle which is in contact with the nerve ending has certain folds. Okay, and you will later realize why these folds are. Okay, and the rest of the muscle is here. Okay, now we need to learn how the electrical signal produced from the neuron is transmitted to the muscle. Okay, since it is the transmission from the neuron to the muscle, it is referred to as neuromuscular transmission. Neuromuscular transmission. Okay, and this will be our topic of discussion today. Okay, so before we move on to the topic of neuromuscular transmission, let us know certain terms. Okay, what do you mean by synapse? Consider this to be my motor neuron, miniature version of my motor neuron. Okay, I have my neuronal end here. Okay, and this new motor neuron is designated to send these signals to a muscle. Okay, so consider a muscle cell here. Okay, in reality, the neuronal end is in close proximity with the muscle, or in other words, the membranes of the neuronal end are in contact with the membranes of the muscular cell. Okay, this region of contact between the membranes of the neuronal end and the muscle cell is referred to as synapse. Okay, but if my goal is to explain you what happens in the synaptic region, or how is this information transferred, I should be able to zoom it, right? So I have zoomed this area and I have drawn it in a larger space. Okay, so this is the neuronal end, this is the muscular end. Okay, in reality they are in close contact with each other, but still there is a minute space in between them. Okay, so the space that exists within the synapse is referred to as our synaptic cleft. Okay, synaptic cleft. Now, in the last video, I told you information from a neuron is transmitted to the other neuron with the help of chemical messengers. And what was that chemical messenger? It was acetylcholine, right, or our ACH. Okay, similarly here too information from the neuron is transmitted to the muscle cell with the help of chemical messengers called acetylcholine. Okay, and this acetylcholine transfers the information from the neuronal end to the muscular end by traveling this distance or traveling the synaptic cleft. So this is an example of chemical synapse because information is transferred from the neuronal end to the muscular end with the help of chemical messengers, and in this case it is acetylcholine. So if you have a question or if someone asks you what is a chemical synapse, you can answer it with the help of our neuromuscular transmission. Okay, now you have another term, the part of the muscle which is in close contact with the neuronal end is referred to as our motor plate. Okay, motor plate, because this is the region where the information from the neuronal end is accepted by the muscular cell and the information is then propagated into the muscle. So this area which receives the information from the neuronal end is referred to as our motor plate. Okay, now we need to learn how acetylcholine is formed. Okay, acetylcholine is composed of two chemicals or two components, acetyl and choline. Okay, from where do we get this choline? Choline is present in our diet. Okay, the food we eat which is rich in protein, this protein is broken down and it is used to form or it is used to produce choline. Okay, but this choline is always in the extracellular space, right, or they are in the blood. This should enter into the neuron. This is our neuron cell. It should enter into the neuron. How does it enter? We have specialized proteins here. Okay, these proteins are called the choline transporters because they transport choline from the extracellular space, that means outside the neuron to inside the neuron. So they have brought in choline. Okay, now we need one more component, that is our acetyl part. And where do we get this? See, the every cell has its own cell organelles, right? That is our mitochondria, endoplasmic reticulum, rough endoplasmic reticulum, smooth endoplasmic reticulum, lysosomes, etc. Okay, and these organelles are for the perfect functioning of the cell, right? So similarly, our neuron also has a mitochondria here. Okay, and what happens in mitochondria? If you recollect from the biochemistry classes, in the mitochondria happens the Krebs cycle, right? And what is the main component of the Krebs cycle? It is acetyl-CoA, right? Acetyl-CoA. So the cell uses the acetyl-CoA from the mitochondria present inside it. It removes the coenzyme part or CoA part and it uses the acetyl part to form our required chemical, that is acetylcholine. Now you have acetyl, you have choline. Now you need to form acetylcholine. It doesn't occur naturally or it doesn't occur spontaneously. There should be the presence of a particular enzyme. Okay, and that enzyme is referred to as choline acetyl transferase. Okay, in the presence of the enzyme choline acetyl transferase, acetyl and choline are used to form our acetylcholine. Okay, that is how our acetylcholine is produced. But is acetylcholine rapidly spread across the neuronal membrane? The answer is no. Okay, this acetylcholine, after their formation, they are stored in specialized bag-like structures. Okay, which are present in the neuronal end. Okay, we have bag-like structures which are also called the vesicles. See, so you have the vesicles and the vesicles have proteins, okay, which help in the transfer of acetylcholine from the neuronal end into the vesicles. So this acetylcholine in the neuronal end enters the vesicles and they are stored in these vesicles which are present in the neuronal end. Okay. So the final structure looks like this. We have our motor neuron, okay, with the neuronal end having acetylcholine. We learned how this acetylcholine is formed. Okay. Now, consider I gave a threshold stimulus to the neuron, okay, and an action potential is created. The action potential is traveling and it reaches the neuronal end. Okay. Do you remember these channels? These are our favorite voltage gated sodium channels. Okay. So the action potential reaches the neuronal end, and as always, it stimulates the opening of the voltage gated sodium channels, and it allows the influx of sodium. Right. There is influx of sodium. Now, what happens when there is influx of sodium? When there is influx of sodium, the membrane potential in this region, okay, starts to become more positive. And what was the membrane potential in the neuron cell? It was around negative 90 or negative 85 millivolt, right? Okay. So as sodium enters, it becomes more and more positive. Okay. And when it reaches around the positive 30 millivolt mark, okay, what happens? There are another set of protein channels here, okay, and these protein channels are called the voltage gated calcium channels. We learned about the voltage gated sodium channels. Here we have the voltage gated calcium channels. Okay, these are the voltage gated calcium channels. They are present all through the neuronal end. Okay, when the potential in the membrane reaches up to the positive 30 millivolt mark, okay, the voltage gated calcium channels open and they allow the influx of calcium from outside the cell to inside the cell. Okay, and what does calcium do, or what role does calcium have? Okay, consider we have two different proteins. Okay, they are the V snare proteins and we have the T snare proteins. Okay, so this V snare proteins, I am zooming a acetylcholine vesicle here. This is acetylcholine vesicle here. Here there is a protein. Okay. Okay. This is the V snare protein. Okay. Now, we told that neuromuscular transmission happens with the help of acetylcholine. Acetylcholine is present within the vesicles. This acetylcholine must be, it should be released out, and this released acetylcholine will be only able to cross the synaptic cleft and reach the muscles, right? And how does that happen? We have our neuronal end here. We also have a certain protein here. Okay, we have a certain protein at the neuronal end. Okay, and when these proteins are zoomed in, it looks like this. Okay, so we have the V-snare protein here, and the proteins present at the neuronal end are referred to as the T-snare proteins. T-snare. Okay, see, these acetylcholine will be able to bind to this, right? See, the V-snare protein will be able to bind to the T-snare protein, right? Okay, and when this happens, this protein part or the attachment part disappears and the acetylcholine part, the vesicular part opens. Okay, so we have the neuronal end here, we have the acetylcholine part, we have the V snare protein, we have the T snare protein. When these proteins bind, this protein part disintegrates and the membrane of the vesicle which contains acetylcholine merges with the neuronal end and it disappears, leading to the opening of the vesicle. Okay, and now acetylcholine will be able to move out, okay, into the synaptic cleft. Okay, so the role of calcium here is to join the V snare protein present in the vesicle of acetylcholine to the T snare protein present in the neuronal end. Okay, so that these acetylcholine present in the vesicles are released out into the synaptic cleft. Okay, this process where the membrane of the vesicle merges with the neuronal end and it releases the acetylcholine outside is referred to as exocytosis. Okay, exo means outside, cytosis means from the cell. Anything taken into the cell is endocytosis. Anything pushed outside the cell is referred to as exocytosis. So, with the process of exocytosis or by the process of exocytosis, which is helped by the V snare and T snare proteins and our calcium ion, exocytosis of the acetylcholine vesicles happens and acetylcholine is released into the synaptic cleft. Okay, now what happens? Which now acetylcholine has been released into the synaptic cleft. Okay, now we need special proteins which are present on the motor plate that are able to receive this acetylcholine, right? So it is like our neuronal end talking and our motor plate or our muscular end listening. Okay, so in order to listen, it needs ears, right? And here the ears are the proteins which are able to bind to this acetylcholine. Okay, so acetylcholine starts to bind with these receptors. Okay, these receptors or these proteins are referred to as our ligand gated ion channels, where our ligand is acetylcholine. Okay, and how are they ion channels? These proteins, they have two functions. Okay, they look exactly like this. Okay, so this is a protein where ions can pass, okay, into the cell and outside the cell. But for this ion channel to open from the inside, it needs a ligand. Okay, and the ligand here is our acetylcholine. Acetylcholine binds with this region. Okay, and what happens? The inside of the channel opens, thus making it a possible way for transmission of ions, okay, or transfer of ions into the cell and outside the cell. Okay, so we have our acetylcholine which will bind with our ligand gated ion channels. Okay, there is one more name to this channel. It is also called the nicotinic receptors, because not only acetylcholine but nicotine also looks exactly like this. So nicotine is also able to bind to these regions. Okay, since this channel can be opened by both acetylcholine and nicotine, it can also be called the acetylcholine receptors, nicotinic receptors, or in a broader term, it can be called the ligand gated ion channel. Okay, so when acetylcholine binds with this ion channel, what happens? This channel opens. Okay, this channel opens. So now what happens? This is another cell. Okay, and it has a resting membrane potential of around you say in the range between 8070 to -90. Okay, can be anything between. So let us take it as -70. Okay, let the resting membrane potential of the muscle cell be -70. Say 70. Now outside every cell, I have told you there is one ion which is in large amounts, and that is sodium. Sodium is in large amounts outside the cell. Okay, when these ion channels open, okay, comparatively or compared to the inside of the cell, sodium is high outside. Okay, and compared to the outside region, the amount of potassium in the cell is high. So what happens when this channel opens? These ions start to move according to their concentration gradient. So this is our channel here. Sodium starts to move out. Sodium starts to move in, and potassium starts to move out. Okay, sodium starts to move in and potassium starts to move out. Whenever there is an influx of an ion into the cell, what does it try to achieve? It tries to achieve its Nernst potential or equilibrium potential that for that particular ion which is being which is entering into the cell, right? So here, which is the ion that is entering into the cell? That is sodium. So sodium will try to achieve its equilibrium potential, and in our earlier classes we have learned that the equilibrium potential for sodium is around positive 65 millivolt. So it will try to achieve that. Okay, so there will be rapid influx of sodium. Rapid means large amounts of sodium influx compared to the amount of potassium that is going out. So if 5 sodium gets in, it is around 2 potassium that moves out. Okay, so in result there will be 3 positivity inside. So similarly, our membrane cell starts to become more positive or they start to depolarize. Okay, and they start to depolarize, and it reaches, and due to the sodium influx, this negative 70 then goes to negative 60, then negative 50, negative 40, negative 30, it reaches 0. Okay, this 0 portion or this 0 millivolt state is referred to as the motor end plate potential, because now due to the influx of sodium through the ligand gated ion channel, we have our motor plate potential to be around 0. So it is referred to as end plate potential or motor end plate potential. Okay, now some sodium that is influxing into the muscle cell moves to the nearby region, and the usual story happens. It triggers the voltage gated sodium channels there. It makes it into the threshold voltage, and action potential is created, and the action potential spreads throughout the muscular cell. Okay, now in the next chapter we learn how this action potential is capable of causing muscle contraction. Okay, so this is how the information or the electrical signals from the neuron is transferred to the muscle. In summary, if you are writing it in points, we can tell first there is an consider an action potential to be created in our neuronal cell. Okay, action potential is created. It travels through the neuronal cell and it causes the depolarization of our neuronal cell. Okay, and during this process, the potential of the cell reaches around positive 30 millivolt, which helps in the opening of voltage gated calcium channels, and these calcium channels open and cause influx of calcium. Okay, influx of calcium, and this calcium, they help in the linking of two proteins. What are they? That is the V-snare protein present on the vesicles of acetylcholine and the T-snare proteins present on the neuronal end. And when these two proteins link, what happens? The protein part disintegrates and acetylcholine diffuses out into the synaptic cleft. So acetylcholine diffuses into synaptic cleft. Okay, by exocytosis. Okay, and this acetylcholine is then taken by the ligand gated ion channel present on the muscular cell or muscle cell. Okay, and what happens when this ligand, that is the acetylcholine, binds to the channel? It opens and causes passage of ions. Which are the ions? Sodium and potassium. Sodium into the cell and potassium outside the cell. Okay, since there is influx of sodium into the cell, it tries to achieve its equilibrium potential, and there is high influx of sodium. So the potential of the muscle cell reaches from resting membrane potential, that is around negative 60, to the end plate potential EPP, that is around 0 millivolt. Okay, now some of the sodium that have influxed into the muscle cell moves into the neighboring regions and causes our depolarization cycle. Okay, this is what you write when you are asked about our neuromuscular transmission or our chemical synapse. Okay, if someone ask you about chemical synapse or neuromuscular transmission, this is what you write. Okay, draw a diagram and write it like this. Okay, now in the next section we will be learning how this action potential that is generated in the muscular cell is able to cause contraction and relaxation of the muscle cells. In the last session, I told you about certain things about our myelination and all, right? Okay, so I have to discuss or I forgot to discuss two clinical conditions that are related to that. Okay, we have myelination in our peripheral nervous system, and we also have myelination in our central nervous system. Okay, the myelination in our peripheral nervous system is what we learned in our last session, and it was due to our Schwann cells, right? It was due to our Schwann cells. Okay, so we have certain neurons which are myelinated. Okay, we have myelination. Okay, there is a condition where our body recognizes this myelination present in our peripheral nervous system to be an intruder or to be a outside person. So our body attacks these myelinations. Okay, and what happens? These myelinations are destroyed, and when these myelinations are destroyed, the myelinated neurons won't be able to transmit electrical signals. Okay, so what happens? The person becomes weak, he cannot move his limb, he might become even paralyzed. Okay, and that condition is referred to as our Gullian Barre syndrome. Okay, sorry for my pronunciation, it is Gullian Barre syndrome. Okay, the Gullian Barre syndrome is a demyelinating disorder which is autoimmune. Autoimmune means our body attacks, it is due to our malfunctioning of our body's immune system. So our body's immune system recognizes this myelination present in the peripheral nervous system to be an outside person or intruder, and it attacks it. Okay, destroying the myelination and thus causing paralysis. What type of paralysis? Ascending paralysis. It starts from the lower limb mostly, and it progresses up. Okay, and when it reaches our thoracic region, it starts to paralyze our nerve supply to the diaphragm, and thus we will be not able to breathe, and the patient goes to respiratory arrest and he dies. Okay, so how is it managed? It is managed by removing that antibodies that attack the antigen. Okay, so that process is called our plasmapheresis, where the autoantibodies present in the human body are taken out by a machine, and fresh blood is free of the antibodies is returned back into our body. Okay, a similar disease which affects the CNS or the demyelinating disorder that happens in the CNS is referred to as multiple sclerosis. Okay, multiple sclerosis. So the myelination in our central nervous system is due to another group of cells. Okay, and they are called the oligodendrocytes. Okay, just for your information. Okay, so that's it from today's session. See you later. Make sure that you stay crunchy, you stay hungry for the next session, and you read about it. Okay, thank you. Have a nice day. Cheers.